The senior citizens, your stem cells are already dormant. Your stem cells SC. When we are 2 years old, the stem cells are very active. But as we grow older, when we reach the age of 60 plus, the stem cells become SC. What is SC? Senior citizen stem cell. Wala nang gusto, ayaw nang kumilos, you would rather just sit down, okay? PHHC. What's PHHC? Philippine Homicide and Housing Committee during the time of the late President Macapagal. Remember that? Oh, National Housing Committee, right? So the job of the stem cell is PHHC. Pahiga-higa kain. Senior citizen kasi. But with the proper stimulus, stem cell enhancing, the senior citizen stem cells can become junior citizen stem cells. So they'll be activated. In young animals, how active are their stem cells? You can cut the liver into two. And within 1 to 3 months, it is totally repaired. No, but I think I'll try that as a senior citizen. What will happen? Senior citizen, you cut the liver into two, what happens? Okay. One of the fundamental properties of the stem cell is that it does not have any tissue-specific structures that allow it to perform specialized function. Nothing to do, just like a baby. No work. Unless it becomes a heart muscle, it contracts. It becomes a liver cell, it detoxifies. It becomes a kidney cell, it removes toxic urine. Okay. For example, a stem cell cannot work with its neighbors to pump blood through the body, like a heart muscle. And it cannot carry oxygen molecules to the bloodstream like a red blood cell. However, unspecialized stem cells can give rise to specialized cells, including heart, muscle, blood cell, nerve cells, and so on. So do you think stem cell technology is good? What do you think? That's the only solution for us, especially when we are already senior citizens. When unspecialized stem cells give rise to specialized cells, the process is called differentiation. When it matures from a baby, that is differentiation. When it becomes specialized or functional. Differentiation of stem cells are through internal signals, through their genes, to internal signals, and external signals brought about by chemicals secreted by other cells, physical contact with neighboring cells, and certain molecules in the microenvironment in between. And it can be done by nutrition. Okay, that it can be enhanced, but you should know what to use to enhance the stem cells. Okay. Resident stem cells, unspecified or undifferentiated cells that provide a source of renewal of skin, identification, isolation, and derivation of stem cells, and blood cells throughout life. Isolated stem cells, unequivocal identification of stem cells requires their separation and purification, usually based on a combination of specific cell surface markers, and that is done in the laboratory. Isolated stem cells, for example, hematopoietic stem cells, can be studied in detail and used in clinical applications such as bone marrow transplantation. Cultured stem cells, that means you harvest the stem cells and culture it so that it multiplies, it can replicate, it can differentiate. It is desirable to culture and expand stem cells in vitro to obtain sufficient quantity for analysis and potential therapeutic use. Resident stem cells, unspecified or undifferentiated, that provide a source of renewal of skin, intestine, and blood cells throughout life. Isolated stem cells, unequivocal identification of stem cells requires the separation and purification usually based on a combination of specific cell surface markers. Isolated stem cells, for example, hematopoietic stem cells, can be studied in detail and used in clinical applications such as bone marrow transplantation. Okay. So here is the totipotential stem cell, the zygote. From the fifth day, it becomes blastocyst with the inner cell mass, okay, which when harvested is a pluripotent stem cell. It can be cultured, embryonic stem cells of culturing, okay, and ultimately it leads to the production of pluripotent somatic stem cells for CNS, okay, PNS, peripheral nervous system, okay, so blood, liver, skin, mesenchyme, etc., multipotent stem cells. Then you also get primitive germ cells that converts into egg and sperm cell. Types of cultured stem cells: embryonic stem cells from a 5-stage blastocyst. Blastocysts are immunostatically isolated inner cell mass called embryoblast from blastocyst. And that is harnessed, this one, and that is cultured. Maintain a stable euploid karyotype, that means its torsion number is 46, even after extensive culture and manipulation. Can differentiate into a variety of cell types in vitro, in the laboratory, and can contribute to all cell types. Okay? That is already in the laboratory. So this is again human developmental, okay, continuum from single cell embryo, 1 day embryo, 5 to 7 day embryo, 6 weeks embryo. From the 5 to 7 day embryo, you get the blastocyst and the embryonic stem cells, which is pluripotential. And from the 6 weeks embryo, you get embryonic germ cells, primordial germ cells, which is pluripotential. And from the fetus is already, when it's already 2 months, it's called a fetus, no? Already formed, already. And then later on, when it is an infant, okay, cord blood stem cells, and then they harness also the placental stem cells from the placenta, which are all pluripotent or multipotent. And then from the adult stem cells, okay, from the skin and other parts, the germ cells, you get the adult stem cells, pluripotent and multipotent, and even the teratocarcinoma, germ cell tumor, and embryonal carcinoma arises. Embryonic germ cells here, gonadal tissue from 5 to 11 week post-fertilization from the embryo or fetus. Okay, here. Same pluripotency as embryonic stem cells, difference the imprinting status of some genes. Its gene is different already from the embryonic stem cells. Here, mesenchymal stem cells, that was the topic yesterday. Okay, mesenchymal stem cell, MS or MC, okay, derived from the bone marrow, muscle, adipose tissue, peripheral blood, and umbilical cord blood. So that's what we got. So here in the Philippines, various hospitals are using different stem cells. Some hospitals, they use the bone marrow, some they use muscle, adipose tissue, some they use peripheral blood, and some umbilical cord. The mesenchymal stem cells can differentiate into mesenchymal cell types, including adipocytes, and that is fat cells, osteocytes, bone cells, chondrocytes, and that is cartilage and myocytes. So over here, mesenchymal stem cells to osteoblasts, to osteocyte, bone, adipocytes, to fat cells, chondrocytes, to cartilage cells. Bone marrow, mononuclear, postnatal muscle and brain. Okay. Multipotent adult stem cells. We get that from bone marrow. Are very rare cells that are present within mesenchymal stem cell cultures from postnatal bone marrow. Can differentiate into various cell lineages of mesodermal, ectodermal, and endodermal origin. That's what was discussed. Okay. Neural cells, brain, from the heart. Your heart is that from the heart, that becomes cardiac muscle. You get it from the bone marrow, that becomes blood cells. Okay. And they're still called multipotent adult stem cells. Neural stem cells, the nerve system, okay, fetal and adult brains, they from subventricular zone, ventricular zone, and hippocampus. The neural stem cells can be cultured as a heterogeneous cell population of monolayer or floating cell clusters called neural spheres. Neural stem cells can differentiate into neuron and glia in vivo and vitro. Okay. So over here, you have the neural stem cells from the brain. It differentiates into neurons. The neurons are responsible for nerve conduction and into astrocytes and oligodendrocytes. These are supporting cells. They supply nutrition for the neurons to function. Medical uses of stem cells. Stem cells technology can be used on the following condition. For patients who had a stroke, traumatic brain injury, learning defects, Alzheimer's disease, Parkinson's disease, baldness, blindness, deafness, missing teeth, wound healing, amyotrophic lateral sclerosis, weakness, paralysis, myocardial infarction, heart attack, muscular dystrophy, degeneration of the muscle, diabetes, multiple sites on cancer and Crohn's disease, bone marrow transplantation, currently established, spinal cord injury, osteoarthritis, rheumatoid arthritis. Current status of stem cell therapy in the Philippines and worldwide. Adult stem cells are currently used in a number of therapies in humans, but they are rare and not yet well defined. Adult tissue stem cells are restricted in the range of cell type they can make. Their exact role, frequency of occurrence, and identity are generally not well understood. Embryonic stem cells can reproduce all tissues of the adult, but are currently hard to control. That means culturing them, you are not sure what will you get. Will you get enough stem cell? Is it? Or will you get a different kind? That's what you are imagining, okay? Dr. Ramon Marquez, Professor Emeritus of the University of the Philippines College of Medicine and academician of the National Academy of Science and Technology, raised in his commentary about the current status of stem cell therapy the following concerns. He asked, is the number needed to treat with benefit compared to the number needed to harm already established? Example, you treated patients with stem cells, about 100 patients. Okay, let's say 50% improved, but 50% deteriorated or they get cancer. That is question. How many percent is good and how many percent is bad, right? Well, it's saying if it is 99%, that must be okay. But until now, they don't know how many percent will make it, how many percent will not make it. Okay? It's not even 50-50, they don't know. Next thing he is asking is, are the colonies of cells harvested from the patient or other donors clarified to be healthier cells that can positively control and improve the impaired function of the diseased organs or tissues of the patient? Remember, you are harvesting stem cells from animals or even from persons and giving that to a sick patient. Can that sick patient, can the stem cells survive in that sick condition, right? Is the technique to harvest the stem cells, there's something wrong with this. Is the technique to harvest the stem cells already standardized and considered the best approach? Is the technique that is existing in the laboratories in different hospitals here, is that already the standard? That is in fine. Okay? Is the best media for culturing the cell colonies already known and determined to be the gold standard? The culturing method is still a big question to him. Okay? Are the colonies of cells harvested from the patient? What are the chances of rejection by the body, since the new cultured cells are being returned to a known hostile or diseased environment? So it is easily rejected. What is the inter and intra-observer variability in determining the success or failure of the procedures? How long is long before repeating any or all of the above steps? So in his mind, it's still a big question mark whether stem cell transplantation will be effective. Okay? Leukocytes. Now this is the current status of stem cell therapy. Here is the mesenchymal stem cells. Okay? Here is the mesenchymal stem cells. What will happen if you introduce a stem cell to another individual, especially from animals? Okay? By the way, if we get the stem cells from our own body, that's called autograft. Autograft, from your own body. Okay? So there is no rejection there because that is from your own cells. Okay? Or if you get that from a twin, it's still no rejection because they have the same DNA. Okay, isograft. If you get that from another person, that is allograft. From one person to another, there is a problem of rejection. And more so if it is cellograft, you get that from an animal and transfer that to a man, that will be more problem. Okay? So look at what will happen.
What about if your brother or sister any lineage in your family?
Allograft pa rin yan. Okay. So look at this. Now this is a stem cell. This is transplanted to various threads of multipotential stromal cell space at the delivery site, leading to loss of cells. So when you transfer these cells, okay, you inject that to the heart, to the liver, or to any damaged organ, okay? So what is the fate of the stem cell? That's the mesenchymal stem cells, okay? They will be invaded, they will be attacked by white blood cell, by macrophages. The white blood cells, the leukocytes, the B cells, will release antibodies to destroy them. The macrophages will eat the stem cell, the stem cell, okay? And what will happen? Leukocytes and macrophages that are brought into the multipotential stromal cells implantation site as part of the non-specific inflammatory response can secrete pro-inflammatory cytokines and chemokines and can increase cellular stress and generate reactive oxygen species. So it will release a lot of inflammatory cytokines. So it will enhance inflammation and could release reactive oxygen species that might activate apoptotic cascades that will lead the stem cells to commit suicide. But that's not the only thing. The inflammation that is caused will cause damage to the recipient. Wound and ischemic sites where the multipotential stromal cells therapy is usually required are low in vascularity, kulang sa blood supply, and a hypoxic regions, which might add to mesenchymal stem cell stress. So the stem cells could die because of this lack of vascularity, and also lack of attachment of multipotential stem cells to the extracellular matrix may cause mesenchymal stem cells to detach, undergo anoikis, okay, cell death, and ultimately lead to cell death. Okay, it cannot attach. The problem is, I was told, I went to see a case whereby a lady, well known, went to Germany, she had stem cell transplant. Okay, I don't know what's the reason, maybe to become younger. But when she came to the Philippines, she had a lot of lesions in the face that could not be handled by even a dermatologist. And another case, I heard from a well-known, respectable person, sabi niya, my compadre went to Germany for the stem cell transplant. In Germany, they use lung. Okay? And he has only a liver cyst. The liver cyst is very common on senior citizens.